Chapter 16 is really all about DNA, the structure of DNA, and how that DNA structure was deciphered. And actually, the first experiments that I want you to know about in their contributions is really to try to figure out what is the genetic material. Is it DNA? Is it protein? And there was really a larger case for protein, just because we know that protein structure is incredibly diverse. DNA is DNA is DNA. It's all made from the same building blocks, whereas protein has 20 different amino acids. DNA only has 4 building blocks, 4 different types of nucleotides. And so these first scientists basically were kind of up against a lot of scrutiny, because more of the scientific community believed that the genetic material was protein. So the first experiment I want you to know is Griffith. And Griffith actually is responsible for doing what's called transformation of bacteria. We're going to actually do a transformation lab next week in class, where you're going to transform bacteria by delivering a new gene to it, a gene from a jellyfish that's going to help it glow in the dark. So Griffith's lab wasn't quite as exciting as that, but he did kill a lot of mice. The first 3 parts of his experiment here were basically the control part of the experiment. And then this step over here is the actual transformation. So what he did is he worked with bacteria, and he had 2 strains. He had an S strain and an R strain. S stands for smooth. And as you can see here, those smooth bacteria actually have a gene that codes for this capsule, the smooth capsule. And so if those bacteria are injected into mice, it actually causes pneumonia. And the population of mice injected with that would die. Then he took a second strain of bacteria, the R, the rough strain, and injecting that into the mice. Notice it does not have a capsule here. Okay. And so those do not cause pneumonia. So notice the mice stay healthy. They don't die. The third part of the experiment, he took these same S cells, okay, except this time he exposed them to heat to kill them. Okay, so injecting these into the population of mice, notice, do not cause pneumonia. So the purpose of really these 3 steps is just to basically support that you must have living S cells, not heat-killed ones, living S cells, in order to cause pneumonia. Okay. So in this very last, very important step, he actually took a mixture. He took these heat-killed S cells, which normally do not cause pneumonia. He took these living R cells, which again, normally by themselves do not cause pneumonia. And he created a mixture of those 2 types of cells, injected it into the mice, and of course we can see the mice die due to pneumonia. And he didn't really know what was actually doing the transformation. He knew that something from this heat-killed S cell, some kind of chemical, is actually moving into these R cells and transforming them, changing them. We know now it's DNA, it's a gene that's actually moving over, but Griffith didn't figure that out. What he did figure out, though, is after these mice died, he analyzed samples from them and found living S cells. So actually, it wasn't until 1944 that Avery, McCarty, and McLeod kind of piggybacked on Griffith's experiment. And so they purified transforming agents, found it to be DNA. They actually looked, is it DNA, is it RNA, is it protein, and found that the only chemical capable of doing transforming was DNA. And so not surprisingly, this didn't really satisfy the scientific community. We usually need, lots of evidence, lots of support before we change our minds. And so it wasn't until 1952 that this experiment, Hershey and Chase, basically concluded the same thing, and that's that DNA is the genetic material. But they did it in a different way, different experiment. They used something called a bacteriophage. So that's this structure here. Bacteriophage is actually a virus. but this virus infects bacteria, and so that's why it says bacteriophage. Okay, and basically the way viruses work is they are made of two things. Okay, they're made of DNA and they're made of protein. Okay, so basically they're made of the two chemicals that we're debating between. And so whatever the genetic material is, okay, must be the chemical that's actually being injected into this bacterial host cell and reprogramming that bacterial cell, basically hijacking it, telling it how to make new viral particles with that genetic material. And so what Hershey and Chase did is they basically thought, if we can trace the protein through a round of infection, and if that protein ends up inside the bacteria, then we know that protein is the genetic material. And so what they did is they basically tagged the protein. You probably remember those disulfide bridges that make up that tertiary structure. Sulfur would be an element that's unique to protein. DNA does not have sulfur in it. And so they tagged that sulfur with a radioactive material shown in pink here and traced it through the infection. Okay, and so here's the actual infection stage between the bacteriophage and the bacteria. Then they used a blender. And so by using the blender, it basically shakes off any viral particles that are outside the cell. Okay, but then notice a chemical from the virus has already been injected into that bacteria during the infection stage. Here you can kind of see it starting to sneak in. Then they used a centrifuge, and a centrifuge basically just spins it really fast. And so the heavy stuff, in this case, the heavy stuff would be bacteria, forms kind of a solid pellet at the bottom of the tube. And then the lighter stuff on the top will be any of these little viral particles that are outside of the cell. And so, again, in labeling the protein, we found that radioactive dye is still in the viral particles. It did not enter the bacteria. And so that rules out protein as the genetic material. And so when they did the same thing with this time labeling DNA, labeling the phosphorus part of the DNA nucleotide. Again, phosphorus would be unique to DNA. Protein does not have phosphorus in it. And so in this case, they're using a radioactive dye shown here in blue to tag the DNA. And not surprisingly, you can kind of see that radioactive dye is what enters the cell. And then when we centrifuge it, it shows that radioactive DNA is in the bacteria. And again, that makes it the genetic material, because we need the genetic material to tell the bacteria how to make new viral particles. So this is really the one that convinced the scientific community, and then all the focus is really going to shift to figuring out what the structure is of DNA. So now there's kind of a race that begins, trying to figure out what the structure is of DNA. And so people started looking back a little bit in time. Chargaff was a biochemist. He's the one that told us that DNA is made of nucleotides. And if you recall, remember, a nucleotide is made up of a phosphate. That's what gives DNA its negative charge. It's made of a pentose, or a 5-carbon sugar. And it's made of one of 4 nitrogen bases. Okay, so remember the nitrogen bases in DNA are A, T, G, and C. Okay. And Chargaff didn't really know how all these nucleotides were pieced together, but what he did find out when he analyzed lots of different samples from lots of different organisms is however much adenine he found, he would find that same amount of thymine. However much guanine he found, it would equal the amount of cytosine. And so we call those Chargaff's rules. If we take a look now at how those nucleotides get pieced together, so notice we have phosphate, sugar, nitrogen base. So phosphate, sugar, nitrogen base, and that just basically repeats to build a polymer. Okay, notice that the sugar is actually in alignment with those nitrogen bases. Okay, and if we look at those nitrogen bases a little closer, okay, you'll notice some of them are just a single ring. Remember, we call those pyrimidines, so that's thymine and cytosine. And then the other two are double ring structures, and remember, those always end in the word 9. And those are purines, that's adenine and guanine. So then there's kind of an interesting story about a key piece of scientific evidence that's really going to help figure out the structure of DNA. There was a lab at King's College in London. That lab is still there. And their expertise dealt with something called X-ray crystallography or X-ray diffraction. And so basically what they did is they took DNA, in this case, and crystallized it, so we have a solid structure, and then they bombarded it with X-ray. And so the photo, the famous photo that was produced, looks like this, and it was produced by Rosalind Franklin. This is her famous photo. And every time you see these little black smudges, it basically means that a part of the DNA, part of that crystalline structure was hit and struck with X-ray. Whereas some of these little white spaces here means that the X-ray was able to pass through the structure of DNA without hitting anything. And so for somebody who works in this field, when they see a picture like this, they'll say, oh, it's a helix. So actually Rosalind's last journal entry at King's College in London was that DNA was either a double or a triple helix, which we know that the main form of DNA is a double helix. There are some triple helixes that do form when DNA is copying, but she was definitely on the right track. And even more importantly, she was able to do some mathematical calculations by hand and actually figure out what the spacing is from one side of the DNA to the other. And so where this photo became really important is that there was a 22-year-old, James Watson, who was involved in this race to figure out the structure of DNA. And Maurice Wilkins was talking with James Watson one day. James Watson was visiting King's College in London, and Wilkins just basically took Rosalind's photo, all her calculations without her permission, and gave them to James Watson. And of course, when he saw it, his jaw dropped. That was really the piece of evidence that he needed, that it was a helical structure. And so he went to work model building. So him and Francis Crick were partners, and they basically used Chargaff's data as well and Rosalind's calculations. They knew how many nanometers across this space should be. And so he figured if he puts a big one with a big one, a purine with a purine, it really was too wide. You know, there's kind of pieces hanging out over the side. If he put a pyrimidine with a pyrimidine, those two small single ring structures, it was too narrow. But if he put a purine with a pyrimidine, it just fit Rosalind's data perfectly.